Hello, everyone. Just wanted to say before I introduce that we find ourselves a few days away from Hispanic Heritage Month. This is a real pivotal period dedicated to reflecting upon and celebrating the profound influences and invaluable contributions of our Hispanic and Latino communities. From the arts and sciences, which we'll hear today, to business and feeding the nation, our impact is deeply woven into the fabric of our society. This month isn't just about recognizing one community, but all communities that make up Hispanic and Latinos. And really to understand the rich tapestry of cultures and histories and stories that have shaped our nation. As we embark on the speaker series this month and as well in October, let us keep in mind the importance of embracing, learning, and appreciating the diverse experiences that come with Hispanic and Latino heritage. And together, we're going to journey through a month that reminds us of the strength found in the unity in all communities and the beautiful diversity of our Hispanic and Latino communities and the potential of every individual story. So with that, let me introduce Dr. Paula Arruda, an assistant professor of nutritional sciences and toxicology and in molecular and cellular biology at University of California, Berkeley. Dr. Arruda's lab is dedicated to the understanding of the regulation of organelle architecture and function on metabolic regulation and metabolic disease. She did her postdoctoral training in the laboratory of Dr. Hoda at Harvard, and where she became a Pew Scholar. Really excited to hear about the research today, and the floor is yours, Dr. Arruda.
Thank you. Thank you very much, Andres, for the introduction. I'll just share my screen here. Can you all see my screen well? Yeah? Okay. So I would like to start by thanking the opportunity to share my work in this community. I'm very excited. As Andre said, I am an assistant professor at UC Berkeley. I started my lab recently, around a year and a half ago. Coming from Harvard, and it's been very exciting to start my own group. So the main focus of my lab is to understand molecular mechanisms that are involved in metabolic regulation, and also mechanisms that are involved in the development of metabolic diseases such as obesity, type 2 diabetes, fatty liver disease, and others. And we approach this paradigm from the perspective of the organelles, the function of the organelles in the cell. So, So we all undergo cycles of fasting and feeding every day, and in general, the ability to survive during times of nutritional scarcity depends on the capacity of the organism to make metabolic adjustments to maintain homeostasis. So throughout evolution, a series of adaptive mechanisms that involve cell-cell communication, organelle with organelle communication, and inter-organ communication have been selected to promote these metabolic adaptive mechanisms to survive during times of changes in the environment. And this regulation or inflexibility in these mechanisms are causing metabolic dysfunctions, such as metabolic diseases such as type 2 diabetes, fatty liver disease, and cardiovascular disease, that are the diseases that we are interested in. And in this context, an important organ that maintains whole metabolism is the liver, or the hepatocytes. So the liver has a very high metabolic activity, secretes lots of proteins and lipids, and it drastically changed its metabolism according to nutrient availability. So for example, in the fed state, after we eat, so the liver stores energy in the for example, store glucose in the form of glycogen, synthesize lipids through process to the pathways called de novo lipogenesis, and synthesizes a lot of protein. And these processes are regulated majoritarily by the hormone insulin. However, in the fasted states, the hepatocyte completely changes and now starts to produce glucose through process of gluconeogenesis and glycogenolysis, and to oxidize fatty acids that comes from the adipose tissue in order to produce energy, synthesize ATP, and also synthesize ketone bodies that are then exported to be used by other tissues in the absence of glucose availability. So, and within the hepatocytes, an important organelle that is involved in this metabolic regulation is the endoplasmic reticulum, which is the organelle that we focus the most in our lab. So the ER is a key part of receiving nutrient signals, and the ER is involved in most of these processes that are just mentioned, because many of the enzymes that are, for example, important for gluconeogenesis or de novo lipogenesis, they are hosted in the ER membrane. And during the diseases like the stress promoted by overnutrition, the ER becomes dysfunctional. So the ER, like going in a little bit more detail, is a very diverse organelle, and both in terms of function and structure. So although it is formed by a single membrane and continuous membrane that is spread throughout the cytosol, it shows distinct subdomains. For example, the ER sheets are preferentially formed by stacks of membranes that are often associated with the ribosomes, forming the rough ER, as we can see here in this electron microscopy. And this subdomain of the ER is primarily involved in protein synthesis, folding, and protein secretion. And the ER is also formed by a network of tubules that we can see here in this fluorescent microscope. Here is a COS7 cells. And in an electron microscopy, most of the times we can see the ER tubules like in a cross section, looking like vesicles. And this domain of the ER is predominantly involved in lipid synthesis, lipid droplet formation, also glucose production, since the glucose 6-phosphatase is found in the membrane of the tubular ER, and is an important reservoir of calcium. So the ER is also not isolated in the cell. There's ER makes also contact sites with almost all other organelles in the cells. For example, here I'm showing the EM with the mitochondria, with the plasma membrane, with the lipid droplets, and with peroxisomes. So this means that the function of ER can also affect other organelles, even through direct contact sites. And these contact sites are important for exchange of metabolites, calcium, lipids, and among others. So in metabolic specialized cells, so the structure of the ER have been studied a lot in cell lines and model organisms. However, how the structure of the ER is maintained in specialized tissues is less understood, but we do know that the morphology of the ER changes a lot according to the metabolic demands of the cells. So here I'm giving some examples. So here is an electron microscopy of an acinar cell, which is in the pancreas, and this cell type is dedicated to secrete our digestive enzymes, so it's a highly secretory cell. And if you look to the cytosol in the cell, you can see that it's full of rough ER sheets organized in these stacks. Similarly, we can see this type of structure in goblet cells that are also highly secretory. In the other end of the spectrum are cells such as the Leydig cell or the adipocytes. So the Leydig cells are specialized in the production of steroid hormones, which are lipid hormones. And if you look to the cytosol of the Leydig cells, as well in adipocytes, you can see that they are predominantly found in the form of ER tubules. And then in the middle, there are cells more pleiotropic, such as the hepatocytes or the enterocytes that I just talked a little bit about, where there are cells that, in addition to secrete a lot of protein, are also heavily involved in lipid metabolism. And more importantly, the hepatocyte, for example, changes a lot the metabolism according with the nutritional state. So the question that we are trying to understand is how changes in nutrient availability or nutritional fluctuations, both in physiology, for example, during cycles of fasting and feeding, and also in pathological conditions such as obesity, how this impact ER architecture organization in the tissue, and how this remodeling of the organelle impact the function and the metabolism of the hepatocytes. And to answer this question, we are using different types of imaging approach, but mostly we use electron microscope approaches such as transmission electron microscopy, and also FIB-SEM, which I'm going to show in the next slides. And more recently, we are also using lattice structure illumination microscopy, as I also am going to share some slides on that. So for the ones that are not familiar with the FIB-SEM, so this technique works by, so you have a tissue volume, and then you put in the microscope, and then first you take a scanning electron micrograph from the surface of the sample of the tissue, and then with a focus ion beam, this surface, this layer is stripped away from the sample, revealing the layer underneath, which is also then also registered by scanning electron microscopy, and it goes like that until the entire volume of the tissue is imaged and the tissue is consumed during the process. So this results in the generation of like around 6,000 to 8,000 pictures, so terabytes of data, but it provides the highest spatial resolution that probably achievable in microscopy, around 2 to 8 nanometers isotropic resolution. And also the advanced FIB-SEM that we've been using, and we have been doing this imaging in collaboration with Sean Shu and Harold Hess from Genelia. Now Sean is actually in Yale. So with this, they did a little bit of a modification in the instrument compared to the commercially available through that's from Zeiss. And in their machine, we can image a very large field of view, as I'm going to show, around 100 microns per 100 microns per 100 microns. So then next, we have to annotate and segment the organelles. And of course, because we have like 6,000, 8,000 images, it would be impossible to annotate by hand. So in order to do this, we are using deep learning, machine learning approaches using convolutional neural network. So here is an example. Just a second. So here is an example of the type of data we are getting. So this is a liver from an animal in a fed state, and we are going in through Z. So you can see the stacks of the ER, and the ER is segmented here in blue. The mitochondria is segmented in purple, and we have small lipid droplets here in yellow. This is very small portion of our data set, this handling. Here also I wanted to share the scale of our segmentation. Again, ER is shown in blue, the mitochondria in purple, and then you can see multiple nuclei. So we have around 17 to 18 cells in this volume. And as you can see, the hepatocytes are very full, packed with the organelles. And then we did similar imaging. We used similar imaging approach with tissues coming from, that was obtained from animals in different nutritional states. So we first focus on comparing liver from lean versus obese mice. So here is the reconstruction of the images that I just showed, and here is the reconstruction of the subcellular organization of hepatocytes from a mouse, obese mouse. Here's the obese model that we use is the leptin deficient mice, for the ones that don't know. So this leptin is a hormone that suppress food intake. So these animals don't have leptin, so they eat all the time and they become morbidly obese, and they accumulate a lot of fat in the liver. So in order to then do quantification analysis, we extracted all the organelle volumes in 5 different full cells, and then we started to compare how obesity changes or impact the organelle organization. So we first we focus on the ER. So here is the reconstruction of the ER from a lean, normal wild-type mice. And as you can appreciate, a lot of the ER is formed as stacks of ER sheets, although we also see some ER tubules in these hepatocytes. But when we did similar analysis with the cells from the obese mice, we saw that there was a marked remodeling in the ER. So the stacks of the ER sheets were markedly decreased, and the ER in this case was mostly organized in the form of ER tubules. So if we compare the amount of ER was comparable in total, like in lean and obese. However, the subdomains of the ER was remodeled. And to quantify these changes in architectural organization, we then resegmented the whole data set, but now segregating ER sheets, parallel stacked ER sheets and ER tubules, we can see here. So the ER sheets are marked in red, whereas the ER tubules are in gray. And then we quantified the ratio between ER sheets and tubules in the whole data set. And we saw that, in fact, this ratio is decreased in obesity. So the next question we ask is, what's the functional impact of such a change in morphology in terms of ER function, and whether we could rescue the ER shape, and what would be the functional impact if we change the ER shape and try to rescue in the obese state, try to make it similar to the lean hepatocyte. So in other words, we ask, can we restore ER structure organization in order to improve metabolism? So to do that, we then turned our attention to the ER shaping proteins. So the ER shape is maintained by the relative abundance of proteins that shape the ER membrane, and this was beautifully described by the work from Tom Rappaport, Gia Volz, and others. And so they have shown that proteins such as the CLIMP63, which is an ER transmembrane protein, that oligomerizes in the lumen of the ER and then maintains the ER membrane apart around 50 nanometers. And other proteins that seems to be involved in the ER sheets organization is this protein called P180 or RRBP1. And they have shown that if you overexpress CLIMP63 in cell lines, this would drive the formation of ER sheets, because basically once this protein is present in the membrane, the shape of the membrane will be in the form of ER sheets. And the ER tubules are shaped by proteins that contains a specific domain that are called reticulum homology domain that contain a short hairpin. So these are this domain here in red. So these are short hairpins that doesn't cross the ER bilayer. So, and therefore, when they are present, they bend the membrane, forming this tubulation. So proteins that contain this reticulum homology domain are the reticulomes. So there are several isoforms, and also proteins called RIPs. So in order to change the ER membrane and induce ER sheets or tubules, we manipulated the expression levels of these proteins in the tissue. So I'll give an example where we targeted the CLIMP63. So here we wanted to do this in vivo to understand the impact in the tissue function, systemic metabolism. So in order to overexpress this protein in the liver of the obese mice, we clone the CLIMP63 in adenovirus, and then we infect the mice, and then the CLIMP63 starts to be overexpressed after 3 days or so of infection, and stay. The expression is maintained around 2 weeks, and so we examine the livers to see whether we could recapitulate what was shown in cell lines, or in other words, whether overexpressing CLIMP63 could drive the ER sheets formation in a tissue of obese mouse that lost this type of structure, right? So we did the same pipeline using FIB-SEM, as I mentioned before. And then we did all the quantifications, and we saw that, yes, when we overexpressed CLIMP63, we saw that in the ER from the obese hepatocytes starts to have now significant amount of ER organized in sheets, as you can see here in red. And here is the quantification. And what was interesting, we were wondering whether these ER sheets would be able to have ribosome bound as a normal rough ER. And we saw that, yes, when we overexpressed the CLIMP63, the sheets that were being proliferated, they were decorated with ribosomes, as you can see here in this zone in this TEM. So then we examined, so the question was, but then what's the functional consequence of inducing this proliferation of ER sheets? And here we did a lot of functional experiments. I would just show a few of them just to have an idea, to show, or like to share an idea what happened. So one type of experiment that we did, we measured the ER folding capacity, and we did this here using a reporter that was developed in our lab, that basically looked to the folding of a protein that is a slow maturation and folding protein called ASGR. And we saw that when we overexpressed CLIMP63, the folding capacity of the ER was increased. And we also measured the impact in lipid and glucose metabolism. And we saw that when we increased the ER sheets, there was an increase in fatty acid oxidation and a decrease in gluconeogenesis. There was also a decrease in de novo lipogenesis. And when we looked systemically, we saw that overexpression of CLIMP63, or in other words, changing the ER shape, had an impact in the whole body systemic metabolism. So here we did a test that we usually do in our lab, where we inject glucose and then measure glucose over time. And obese mice, they are glucose intolerant. So once you inject glucose, the glucose increases in the blood, and then it's maintained high because there is insulin resistance, and the glucose cannot be uptaken into the tissues. But when we overexpress CLIMP63, we saw that this the glucose intolerance was improved. So these animals were able to uptake glucose faster compared to the controls. And we also saw, when we looked to the livers of the obese mice, that the amount of lipid accumulation here, the lipids are these white circles that accumulate in the cells. And we saw that when we overexpress CLIMP63, there was less lipid accumulation in the cells, which would agree with the fact that we see higher beta oxidation and lower de novo lipogenesis. So we published this work last year. So if you wanted to see more details, you can probably go and check it in the paper. And oh, I just forgot, there was one more experiment that we did. So here what we asked is, can we do the reverse? So what I just showed to you is we induced the ER sheets proliferation in the obese, but now we ask, can we then turn the ER to ER tubules in a lean, healthy mice? And if we do this, can we recapitulate some of the aspects that, or some of the alterations in metabolism that we see in obese mice? So here, in order to induce the proliferation of the ER tubules, we used these proteins that I mentioned, RIP5 and reticulon, and we used the same strategy where we cloned these proteins in adenovirus and then delivered it to the mice. And we saw that when we, so here I'll show the example when we overexpress the RIP5. So here are the controls, and we see that there are lots of parallel organized ER sheets. You can see through the EMs. But when we overexpressed the RIP5, there was a huge proliferation of ER tubules. I put it here, you can see in more details. And the tubules, you can see there's small vesicles in a cross section. And what was interesting is that we saw that in this in the middle of these ER tubules, we see it start accumulation of lipid droplets that are these circles here. And then we measured these lipid droplets by other methods. For example, this is H&E staining, where you can see these white circles accumulated in the hepatocytes when we induce the ER tubules. We also measured the triglyceride amount. This was increased. And we think that this was this paper here published in Current Biology did similar experiments, but in cells. And what they show is that when you have the membrane of the ER curved, this facilitates the nucleation of the triglycerides in the membrane and decrease the energy that is needed for the budding of the lipid droplet. So we think that by forcing the ER to be in a tubular form, this will facilitate the formation of lipid droplets, and that's why probably the lipid droplet starts to accumulate in the cells. So in summary of this work, first we developed this workflow using FIB-SEM and automated segmentation, as I mentioned, in collaboration with Shu from Genelia, which allowed the precise determination of organelle structure in the native tissue. And we also saw that obesity, there is a marked transition in the ER subdomains from ER sheets to tubules, and when we change this conformation, this has an important impact in the metabolism of the cells. So more recently, we are interested, so until now, what I showed to you is an impact of a pathological condition in the ER morphology and function. We now more interested in to understand whether ER remodeling is part of metabolic adaptation to physiological changes in nutritional states, for example, during transitions between fed and the fasted state. So we are now using the similar approach. So we have animals, lean and obese mice, now either fed ad libitum or that we fast overnight. And then we are looking to what happens with the subcellular organization or the organelle structure and communication. So here, again, as I showed now multiple times, this is a TEM of a liver section. So you can see all the ER sheets, the mitochondria here, the lipid droplets, and the nucleus. And when we fast the animals overnight, we see that there is also an important remodeling of the organelles. The first thing we see is that the mitochondria are bigger and more elongated in the fasted state in a hepatocyte. So here is the reconstruction of all the mitochondria in the hepatocyte. So here in the fed state, they are more round. as you can see here, but in the fasted states, they become more elongated, and the number of mitochondria decreases in the fasted state, which suggests that these mitochondria are undergoing fusion process, which has been shown also in cell lines, that in the fasted state, the fusion of the mitochondria allows for this mitochondria to have a better or more efficient fatty acid oxidation in this condition. We also saw that the morphology of the mitochondria was much more heterogeneous in the fasted state, as you can see here, all these different formats, and some of the mitochondria was really engulfing the lipid droplets. So you can see this is the reconstruction of the small portion of the video. The lipid is this dark circle that's being here, you can see in yellow, and then you can see that the mitochondria is really around the lipid droplet. So there's a lot more of this type of structural forms in the fasted hepatocyte. But it was not only the mitochondria that changed the shape during fasting. We also observed that the ER sheets, which are organized in these parallel stacks here, they change and they surround the mitochondria. They are around the mitochondria perimeter, and I hope you can see this here in this image. So when we looked more closer to this organelle interactions, we see that the ER that was around the mitochondria in the fasted state is the rough ER, because we can see the ribosomes are attached to the ER. And this is quite different from these membrane ER mitochondria contact sites that are also known as the MEMS, where the ER is very, very close to mitochondria, like around 10 to 25 nanometers apart, and this is formed by protein-protein interaction. Here, with the rough ER, it's, the interaction between these organelles are the distance is much larger, around 25 to 80 nanometers, as you can see here. And this has been shown also to exist in other contexts, in other cell types. So here is the FIB-SEM reconstruction of the ER in the fed and the fasted condition. And I hope in this video you can appreciate that the ER here in white. So if you delete the mitochondria, yes, you can see that the ER forms this scaffold involving or like enveloping the mitochondria. And here is just one mitochondria that we selected where we reconstructed the ER. So you can see that the ER really wraps or envelope the entire mitochondrial volume. So if you look in 2D, this is what you see here, the mitochondria and the ER, but in 3D, this is how it looks like. So then we developed a pipeline to quantify this in our like very big data set. And as we've shown here, so basically we labeled the ER and the mitochondria, and then calculate the amount of the interactions in the different distances. And we know that the distance between the rough ER and mitochondria is around 25 to 80. So with this type of quantification, we were able to see that in fact fasting induces this type of organelle interaction. And we did a time course. So the data I showed to you before, it was like after like 16 hours fasting, but here we did a time course in different times of fasting, and we see that the fasting time, the amount of the this remodeling of the rough ER around the mitochondria occurs proportionally with the fasting time. But now, so as you know, so with EMs, we have only static images. Although we did a time course, you need to fix the animals, and then it's only like at that time point. And we wanted to see more in more dynamic way, how the ER remodels in response to nutrient availability. So we are also now complementing our imaging with EMs with fluorescence microscopy, and here we are using the lattice SIM with the ELIRA that we acquired in our lab. So here, this is a COS7 cells. It's a cell line among a kidney cell, where you can see very beautifully the ER network. And now what we are doing is trying to image how they are change the shape in response to fasting, feeding, different hormones, different nutrients. And I hope in the next seminar I can show the results we obtain with this approach. And with that, I wanted to acknowledge, thanks to the lab or the people that are doing this work. So this is my recently formed group. And most of the work I showed to you today, I did together with Gunesh Parlakpul. So we worked together during our postdoc at Harvard with Gökhan Hotamishligil, and we continue work together at Berkeley. Here also our collaborators, Gökhan, Sean, Song, and Harold, that we do the FIB-SEM together, and our funding. And I'm happy to answer any question that you may have. And thank you for your attention.
It was a wonderful talk. We do have a question in the chat for you already.
Should I stop sharing now, or
just in case, if you need to go back to the slides, you can keep sharing, or if you want to stop sharing,
I can keep sharing. Yeah. Okay.
Can you elucidate if ER dysfunction is expressed in ciliopathies related to obesity?
Sorry, in what?
In ciliopathies.
Okay. If ER dysfunction, so I don't know. I don't think there is any evidence for that in this context.
I think Dr. Hinton has a question. Go ahead.
Yes.
Hi, how are you doing? It's
nice to see you.
Nice to see you as well. I have a question around the multiple contact sites. So I know that you've shown mitochondria ER, and you're familiar with Luca Pellegrini's work where he's shown wrapper ER in the liver.
Yes.
So I'm wondering if you see some of the same type of concepts, but in the context of other organelles interacting with your facet, say, besides mito ER contact changes.
Like, for example, peroxisome and...
Exactly.
Yeah. So we haven't quantified the peroxisome mitochondria ER interactions, and but we could do it because we have it segmented.
Right.
But we haven't done that. We did only mitochondria, lipid droplets, and mitochondria with the ER, with the different rough ER versus different distances. But it's likely that with the peroxisome, because peroxisome is also involved in fatty acid oxidation in the fasted state. So
it's very interesting. Yeah. So, but that's my only question for now. I'll follow up later for things. But okay. It's a great talk.
Thank you. Thank you.
We have another question. Does the volume of the ER change? In pathology, lipids increase, and in fasting, mitochondria increase. Does this affect the volume of the ER? And does this may this affect its functionality somehow?
Yes, so in terms of the ER, with the obese condition, we don't see a change in total volume. The total volume is same. It's just the shape of the membranes that are different. They change to sheets to tubules. And with the functionality, yes. So we know in obese, there are several aspects of the ER that are dysfunctional. For example, calcium homeostasis and calcium storage in the ER is lower in obese condition. and then which leads to higher cytosolic calcium. And this is a problem because calcium is very important for the function of chaperones inside of the ER. So this relates also to unfolded proteins and activation of the unfolded protein response, which happens in obesity. And there are multiple aspects of ER that is dysfunction, but is dysfunctional in obese, but whether and how they are linked to the morphology, we are exploring. So one thing we know is that if we change the morphology, we can affect protein folding and secretion, also lipid droplets formation, as I showed. The other aspects we don't know yet, like, for example, the calcium dysregulation, the unfolded protein response, whether this is also related to shape. That is not, we don't know yet.
When looking at the dynamics, right, that's what you're proposing next, is it possible that down the road you can have the model where you have your morbidly obese mice, but then they end up losing weight, and how that kind of like pulsing effect of morbid obesity, but then like similarly in humans where they have like gastric bypass, or with the new like Ozempic and things like that, where you end up having a transient expression of high fat
Exactly. So
accumulation.
that's a question I get all the time, whether these changes that we see are reversible. And I think that is possibly reversible because it's related to probably the body weight and the accumulation of lipids in the liver. But experimentally, we haven't done that yet. Like whether, like, for example, inducing or treating the animals with the GLP-1 agonist, how this would influence and how the dynamic. That would be nice also to do in fluorescent microscopy, like when treating the cells and seeing how the changes, how their morphology responds to these drugs.
Do we have any other questions? If not, let's thank Dr. Arruda for an amazing talk.
Thank you. Thank you very much. Thank you all. And if you have any more questions in future, just contact me. I'm happy to share more details of our work. Thank you.
Thank you so much.
Thank you.